Alright guys, welcome back to HIBC2. Today we're going to talk about how to use JMP to plot some of the data for your plant transpiration poster. And to quote my current favorite game developer, the Stellaris developing team, if a picture's worth 1,000 words, then I'm going to educate you at 30,000 words per second. So let's go. Alright, so on all student access, we have here your sample data sets from lab that you collected and will be updating very shortly, I hope. So we have our stomata data here where you're going to put your densities and your pore lengths, and this will auto-calculate your pore area for you. And then in your week 2 experiments, you all did some kind of condition where you measured transpiration. So please give these meaningful conditions up here based on your experiment so that others can properly plot your data. You might have to change the layout of that. So what we want to plot that I'm going to focus on here are stomatal density versus pore size as your first plot, and then your second plot will be tracking the changes of these data sets, so your week 2 data sets and your other groups over time or between conditions for the same plants. And that's called the parallel plot. So we're going to start with the stomata data. In all student access, I encourage you to go through and do this on your own. We have these simulated poster prep data that you can practice with. So here we have some stomatal density versus pore size data. On this other page, we have plants treated with different controls. And notice that these are matched data in the sense that this is one plant with 2 measurements of it. So we're just going to move this into JMP. So we're going to copy it onto our clipboard. We're going to open JMP. I'm using Windows, but all of these buttons that are here on Windows would just be on your top bar in Mac. I'm going to open a new one, Control N or File New, and then I'm going to paste them here. But I'm going to use the Paste with Column Names button so that my column names will get posted up here instead of as the first line of the table. As always, once you put stuff into JMP, you want to make sure your data types are correct. So species, character, nominal, because these are text. And then your raw data should be numeric continuous. Make sure those are checked. All right, so once you're into JMP, you have it formatted like this. This mimics what's going to be on Canvas or in the student access. You are ready to go. So we're going to just graph this, graph builder. We're going to put density on one axis, pore on the other axis. I'm going to turn off this wiggly line. And so something we haven't talked about is how to overlay multiple data sets. So we can either use the overlay species, or rather overlay, or we could use color. Functionally, they do the same thing here. Overlay's a little better because if you were to try to do like curve fitting, overlay will treat everything as separate data sets, whereas color will treat everything as one data set. So I prefer overlay. Once you have it in here, you can resize this if you need to, but you can actually click on your species, your legend over here to select different species. So if we look at the Alencinia, we can see its distribution. We can look at the Jordanicus, the Fahardinium, and of course the Marcelium, everyone's favorite, just rolls off the tongue. And then once you're this far, you guys should know how to save, export, whatever. You can click done. You could screen capture this if you wanted to. You could do your file, export. I guess it's save as on Windows. All right, so that's pore size versus stomatal density. Now we want to talk about how to plot these different experimental conditions in a way that's visually usable. So we have a couple options here. As always, first we have to get into JMP. So we'll copy it from our data set. We'll paste it into JMP with our column names. Again, we check that these are continuous and that this is nominal character. This is all good to go. So the first way you could do this, and is probably the easiest way, is you could just plot these on your y-axis by species and turn this into a bar chart. Because this data set has like 50 species here, it's pretty cluttered. But feasibly, you could do this for your own species. And you can see here that the green bars are all larger than the blue and red bars. All right, so that's one option. The other option, just a little fancier, is called the parallel plot. So this is kind of similar. We just do analyze or graph parallel plot. We just drag our y-axes up here. We won't do anything with species, so we'll leave that alone for right now. We're going to check this scale uniformly. This plots them all on the same scale, which is to say it puts a y-axis on there. I don't know why that's not the default, but here we are. This is the world we live in. By default, you get this kind of chaotic black graph. You can see the trends here, but it's not clear what's happening, so we're going to add a legend to this. And the easiest way to do this is we're just going to right-click it. We're going to go to Row Legend. And then we're going to make sure the species is clicked. We don't actually have to check anything else. We could change the colors to scheme if we wanted to. We hit OK, and bam, now we can see everything. So we can again look at the Alencinia, the Fahardinium, the Jordanicus, which does not really do much. It's so boring. And of course, the Marcelium. Look at that, the Marcelium versus the Jordanicus. What a champ. And again, you can file, save as, or export, depending on if you're Mac or PC, to save your data. Now, obviously, your legend is only going to have 2 or 3 species here, so it's not going to trail off the page. If anyone figures out how to fix this trailing off the page thing, I will be extremely impressed. And with that, we are good with how to make these 2 types of plots. Also, make sure to watch the ImageJ video. We're going to talk about how to quickly and easily analyze, annotate your findings for your microscopy images. All right, thanks everybody, and stay healthy.